They're what? Yeah, so they're looking at different parts of the overall pathway. So today we're going to be looking at the transduction pathways and the responses that we get from those transduction pathways. What Samir just said is actually an important thing to remember when you're going through these. It's like a zoomed in, they're all zoomed in pictures of each other. Essentially what they did in your book is they took an overall pathway and then we broke down the individual pieces. So here's the signaling part, here's the transduction part, and here's the response part. So we're just kind of jumping into specific parts of the pathway. So before we start getting into like the actual diagrams themselves, here are some things that we need to know. So transduction is where we're going to start. And in transduction, what we're doing is we're taking that original signal that came from outside of the cell, whenever the ligand bound to the receptor, all the stuff we talked about yesterday, and we're going to take that initial signal and we're going to spread it through the entire cell in this big cascade, which is going to allow us to amplify the signal and allow us to have multiple checkpoints where the signal can be regulated. And we can turn on and off the overall process. So transduction involves... Is this visible? Yes. Scoot over here. So it involves relaying the original signal induced by the binding. I'm probably off the screen now. Induced. Yeah. So like elicited from. So induced by the binding of the ligand. Throughout the cell. Through a series of complex intermediates. Kind of out of the room. So we are taking that initial signal and then we are going to spread it through the cell and we are going to hit a series of intermediates, meaning this thing is going to activate this thing, which then activates this thing. It's like a big game of mousetrap, essentially. And what this does is this is going to result, so it results in a specific cellular response. So at the end of one of these pathways, there's some kind of goal, there's a target. So maybe we need to produce a bunch of copies of a particular protein because you just ate a certain type of food and now there's a whole bunch of, say, lactase. Maybe you just ate a bunch of dairy, there's a whole bunch of lactase sugar floating around in your bloodstream. It's now time to make a bunch of lactose. There's going to be a signaling pathway that tells your cells in the small intestine, for instance, that, hey, there's a whole bunch of lactose out here. We need to break it down. And that pathway is then going to start causing us to express the gene that produces lactase, and then we will have a way of breaking down that milk sugar. So there's always some kind of end goal with all of these transduction pathways. Now, there are two major advantages to how complex these things are. So, advantages to complexity. There are two. One, you have a greater opportunity for amplification. Meaning that you have a chance every step along the way to activate more and more and more and more signaling molecules that are going to cause the signal to spread out further throughout the cell, and you will be able to generate a greater response with a small initial stimulus. And the second is that you have a greater opportunity for regulation. If you want to turn off one of these pathways, there's going to be a lot of steps along the way where you could shut it down at. And depending on what pathway it is, maybe you want half of the pathway to continue to perform some sort of response, but you need to cut it off at a different choke point to prevent a different cellular response. There's going to be a lot of steps along the way where you can where the cells are able to tailor the cellular responses based on what kind of conditions are going on inside and outside of the cell. Okay, so let's go ahead and contain this thing. So section this off, because we're going to need that space below it. The first thing we're going to look at is the phosphorylation cascade. So this is one of the signal transduction pathways that we see, and in order to talk about that, we need to talk about just in general what phosphorylation is. So I'm going to bring that wall straight down. And we're going to talk about phosphorylation in this little box right up here. Okay. So phosphorylation and dephosphorylation . They are one of the primary mechanisms for transduction. I'm just going to state ST for signal transduction. And there are two major proteins that are involved in doing this. Remember that phosphorylation is just the addition of a phosphate group to something. Dephosphorylation would be the removal of a phosphate group from something. So we have two major enzymes that are involved here. We have the kinase, and this is going to be an enzyme that transfers a phosphate group from ATP to a protein. Yes. You're okay. MEC for mechanism, sorry. I'm abbreviating things. So a kinase is an enzyme which transfers a phosphate. It's going to do phosph from ATP to protein. Okay. And then there is something that removes them. So if there's something that can phosphorylate something, we also have to be able to dephosphorylate in order to shut down a pathway or to reset a pathway. And this is going to be the phosphatase. Phosphatase. This is an enzyme which removes a phosphate group from a protein. Okay. And one of the things that we see is that if you phosphorylate a protein, it tends to activate it. If you dephosphorylate a protein, it tends to deactivate it. And that's where these phosphorylation cascades are going to come in. So kinase equals activation. Whereas phosphatase equals deactivation. And when we say activation and deactivation, remember that's all just shape changes. So when you phosphorylate something, especially a protein, When something binds to a protein, the protein changes its shape. Change in shape equals a change in function. So whenever that phosphate group is added, protein changes its shape to a shape where it can now interact with something new and perform a new function, and we would consider it activated at that point. You remove that phosphate group, it's going to reform that original structure, which does not allow it to activate, which doesn't allow it to interact with the next thing within the step. So where we would see this happening, first of all, this phosphatase, it's right here. You can see the PP, where the phosphate group is coming off of things and making it inactive. Okay, that's where these things are used. So we see them in a couple of places here. And just to make a little key for our diagram, PP equals the phosphatase. Okay, in a phosphorylation cascade, what's going to occur is we will have our original signal being taken up by the receptor, just like we talked about yesterday. That receptor will undergo a shape change, which allows it to perform some sort of action on the first relay protein, which is going to be the initial molecule that kicks off a transduction pathway. And then we're going to have a series of kinases adding phosphate groups to each other. So kinase 1 would become activated, meaning it would have a phosphate group added to it. And then that is going to allow kinase 1 to then transfer that phosphate group to kinase 2, which then allows kinase 2 to transfer that phosphate group to kinase 3, and so on in a cascade all the way down until finally you're going to phosphorylate a protein that is going to perform some sort of cellular response. So it doesn't really matter what that response is. We're just kind of speaking in generalities, but the phosphorylation cascade itself is going to be just passing phosphate groups all the way down a series of proteins, a series of kinases. And one thing I want to add here where it says phosphorylation cascade is this is where we can get a lot of regulation and amplification. So regulation and amplification is possible throughout the cascade. So any of these steps in between the original signal and the final response, we could shut down the process at that point and regulate this cycle, regulate this process. Okay. So that is one transduction pathway, is using phosphorylation. The other one that your book gets into is using what they called secondary messengers. And these secondary messengers are going to be kind of like ligands within the cell. So they're not going to be proteins. And we're going to use this space here, this kind of little T that we've set up for this part. Okay, so second messengers. All right, things about second messengers. They are small. This is going to sound a lot like a definition of a ligand. They are water soluble. And they are not proteins. So small, water-soluble, non-protein molecules. Which participate within these transduction pathways. I like to think of them as being intra or within intracellular ligands. They're kind of like ligands. What's that? They're kind of like ligands in that they are going to be interacting with proteins within the cell, but they're not proteins themselves. But whenever they're binding to these proteins, they're initiating some kind of response within the protein due to that shape change that they bring about. And there were two major second messengers that your book covered. There was the CAMP, so the cyclic adenosine monophosphate. So there's ATP, there's ADP, and then there's AMP, meaning just one phosphate group, right? Adenosine monophosphate. Well, cyclic adenosine monophosphate just has a slightly different structure, and it is used as a messenger molecule within these signal transduction pathways. And I'll show you what that looks like here in just a second. All right, is everyone caught up to here? So the first one we're going to look at is in this very first box here. And this is focused right here on the CAMP, the cyclic adenosine monophosphate. Okay, so lowercase c, uppercase AMP. This stands for cyclic adenosine monophosphate. So one phosphate group on the end of this thing. Is a second messenger. And it is formed through the breakdown. So here we can just do breakdown. So it's formed through the breakdown of ATP. And this is done by a particular enzyme, and we'll put here what the name of that enzyme is. Oh, it's actually right here. The adenyl cyclase. Did I spell that right? That's right there. The reason I left a space here?
Like the left, right under.
Right here? Nope, I didn't. I just, whenever I put a colon, I was just trying to block that. I don't know. It's a formatting preference. Yeah. As long as just leave some space over here. Okay. So this is one of these second messengers, and basically, I feel like it seems so much more complicated than it is. Think of it exactly as being just another protein within this cascade, except it's not a protein. So it's going to be a different non-protein molecule. So essentially what we see happening is, so I'm just going to put our 1 here, because this is where it's occurring. But basically what's going to happen is we would have, in this case it's a G protein pathway, but it wouldn't have to be. This could be within a phosphorylation cascade. One of the steps in between, one of the intermediaries, is going to be ATP being broken down into AMP. So that just means it had some phosphate groups removed. How many phosphate groups would have been removed? 2. Yeah. Smaller than proteins. So essentially that's what they typically mean, would just be smaller than a protein. Because proteins are super huge, right? So just not as big as a protein. Let's see. Okay, so here we have the G protein activating the enzyme that then breaks ATP down into cyclic AMP, and then that would be used to phosphorylate, because it still has a phosphate group on there. That's going to be used to phosphorylate another kinase, which would then go on to maybe perform a phosphorylation cascade. Or maybe the kinase would go straight into generating some sort of cellular response. Essentially, this is just going to be another middleman within one of these processes. So don't make this harder than it is. It's It's a separate messenger outside of proteins. And what I want to emphasize here is that CAMP can be synthesized rapidly and greatly amplify. I don't know if I'm using the word greatly correctly here. Greatly amplify the signal. That is my shorthand for within the pathway. And we see these not only in the G protein pathways, but also in the tyrosine pathways that we talked about yesterday. So this could be in G protein or tyrosine receptors. So I didn't want you to see this diagram and think, okay, this only happens with G proteins. No, it can happen with, it doesn't really matter what the original message is being received by. Second messengers are used all the time within these pathways. Okay. The reason that this can be so good at amplifying a signal is because since we have an enzyme creating this cyclic AMP, remember that enzymes are able to very, very quickly generate product, and they don't get consumed within the project that they're generating. If you have a phosphorylation cascade, you have one protein which then passes one phosphate group to another protein. Okay? Amplification is kind of limited. With this, we can just start cranking out tons of cyclic AMP depending on how much ATP is broken down, and that can just send out tons of secondary messengers to activate more pathways. So this can generate a really big amplification of the original signal. Okay. So the other second messenger that your book highlights is calcium ions. So I'm going to come over here and we'll do number 2. And this is going to be the diagram directly below the one we just did. Calcium is Ca2+, and it is an ion with a +2 positive charge. Okay, so this is used even more than cyclic AMP. So this is used in tons of pathways. So changes in calcium ion concentrations regulate many processes within cells. Not just human cells or mammalian cells, but bacterial cells, all sorts of cells. So most organisms use calcium in their regulation processes. Now, it's kind of weird how your cells store this calcium. So Ca2+ is pumped into the ER, so endoplasmic reticulum, and mitochondria, comma, and out of the cytoplasm. So what this does is it causes the cytoplasm of the cell to have a really low calcium concentration, because all the calcium's either within the membrane of the endoplasmic reticulum or within the membrane of the mitochondria. And then whatever calcium is not stored in there gets pumped out of the cell. So the cytoplasm will always have a really, really low calcium concentration. And that way, if the calcium concentration changes within a cell, then that can be used to set off a series of events as second messengers, just like the cyclic AMP was. We're going to continue point 2 right here. So 2 continued. Oh man, I'm going to pick the face here. Okay, so many pathways. Lead to the release of calcium ions. And this is going to be released from the endoplasmic reticulum or from the mitochondria. And this is going to be done, I guess I shouldn't end the sentence there, release of calcium ions via a different secondary messenger, which your book mentioned. So a different second second messenger, which was IP3. And we'll look at the schematic here in a second, and this will make more sense. This is going to cause an increase in cytoplasmic calcium. So, increase. Cytoplasmic calcium activates other proteins. Which leads to a cellular response. Okay, so if we look at the actual schematic, we can kind of put all of this together. It's really not much different than what we just looked at with CAMP. Remember that this is not a protein, but it's kind of acting like a protein within these pathways because it's just activating new things. So if we look down here, we can put number 2 kind of right here because we see it happening in 2 different spots, here and here. Okay, so again, you would have a ligand binding to an initial receptor. This is using a G protein pathway, but it wouldn't have to be a G protein pathway. Then that G protein pathway is going to set off a series of events where we have a enzyme creating the IP3. You don't need to know why that happens. The IP3 is then going to go and it's going to latch in to an ion channel here. So this would be an example of one of these gated ion channels. And it is going to cause a shape change in the ion channel, which opens it up. Calcium will flood into the cytoplasm. So now there's going to be an increase in cytoplasmic calcium. And this calcium can now go on and generate a bunch of cellular responses. Why would you need to maintain, why would calcium levels need to be really low in the cell in order to use this pathway? Yeah, so like imagine if you already had a whole bunch of calcium in here, then all of the proteins that are going to be activated by the calcium would always be activated. Does that make sense? There has to be a change in the calcium concentration in order to activate these proteins to generate the cellular response. So those are the 2 examples that your book used for these second messengers, which I could see as being one of the more confusing parts of this chapter. Okay. Let's see. Let's move here to the response section. We are nearing the end. That was the bulk. So the response diagram is here to the left. Okay, so at the end of these pathways, we're going to generate some sort of specific response. So at the end, of a transduction pathway, a specific cellular response is generated. And this could be within the cytoplasm or within the nucleus. The diagram that we're going to work off of here is focusing on the nucleus, but it could be all sorts of responses within the cytoplasm. Your book goes through a couple different specific examples, but I'm going to focus on the gene expression aspect. Okay, so this diagram here, we're going to go just below it. Many pathways. Are used in gene expression. regulation. And what that means is the cell is going to produce specific proteins based on the original stimulus from outside of the cell. So based on the original external stimulus. So again, we're being vague, we're being broad because we want to focus on overall concepts, not on specific examples. The examples are useful, but we don't want to get too specific, so you can apply this stuff through multiple contexts. So what we see here is going to be, again, we have an initial stimulus, whatever it might be. Then our transduction pathway here is going to be a phosphorylation cascade, which we just talked about how that occurs up here. So we're going to have kinases transferring phosphate groups to each other. And then finally, that's going to end with some sort of activated protein entering the nucleus from the cytoplasm, and it's going to be activating a protein called a transcription factor. That transcription factor is then going to latch on to a specific region of coding DNA, so a gene, and it's going to latch on to the very beginning of that gene, and it's going to basically recruit the RNA polymerase, the enzyme that actually reads this gene to make messenger RNA. It latches on and basically recruits it to bind to the beginning of the gene and read until it reaches a termination sequence. So this is how your cells know what proteins to make at any given time, is through these signal transduction pathways. So I just want to go ahead and define what a transcription factor is, because your book mentions it a few times and it's included in this. So transcription factors. A transcription factor is a protein which, when activated, bind to a particular DNA sequence, or you could say gene, and facilitates transcription. Which, if you don't remember what transcription is, that's going from DNA to messenger RNA. It is the first step in making a protein. So it's going from the DNA into the messenger molecule that's going to leave the nucleus and go find a ribosome to be built into a protein. Okay. We have one section left. So any questions on the response side? And we focused here on gene regulation and gene expression, but it could also be we need to release glucose from the liver, or all sorts of other cellular responses. It doesn't have to be gene expression. Okay, good. Okay, last section here. This is out of 11.4, and it's the end of 11.4. And the first thing we're going to look at is signal amplification. So as transduction pathway progresses, the number of activated molecules increases at each step. This occurs because the proteins that are activating these molecules exist long enough to generate more molecules than what are currently active. What this does is it increases cell response with one original stimulus. And if we were to kind of sketch how this would occur, maybe you have that original stimulus activating one other molecule, and that one new molecule can activate two different ones, and then those two are able to maybe activate, a few others. And eventually we have a lot more activated molecules than what we started with. And by the time we get down to the response stage, we have a really big response from a small original stimulus. And this is able to be done due to the complexity of these pathways. And the very last thing is going to be down here in this bottom right corner, the specificity of cell signaling. So how can we release one hormone, say epinephrine, and generate so many different responses throughout our body if that epinephrine, that adrenaline, is entering our bloodstream? So we're able to get a bunch of different physiological changes from the release of one signaling molecule. And the reason why that occurs, so first of all, let me set that up. So the same ligand is able to elicit or generate responses from many different types of cells. Or even within the same cell, you can get a bunch of different responses that have nothing really to do with each other. And this is possible due to pathways containing a specific set of relay molecules, whether it's proteins or second messengers. So for instance, with adrenaline's going to always bind to the same receptor. But depending on the different proteins within that transduction pathway, we will end up at a different spot. It's kind of like if you were to turn down a road and you had like 5 different options of where to go from that one single road. Depending on the pathway you take, you're going to end up at a different location. You'll end up at a different response. So these metabolic pathways have a whole bunch of different roadways or series of proteins that the signal can be spread through to get you to a different cellular response. And it's all based on this lock and key mechanism, where you can see in this diagram where you have different possibilities for how the signal is going to be transmitted through the cell. Okay, so that is